Let's talk about the nucleus. Okay, so we all know the nucleus is the largest cell organelle, and this is the distinct cell organelle. We otherwise call it as brain of the cell because it contains the blueprint of the cell, none other than the genetic material. And it mainly controls all the activities of the cell. That's why we call it as brain of the cell. It was first discovered by Robert Brown in the year 1931. Robert Brown discovered this. So in the first he have observed in orchid cells. So later the material was stained with basic dyes, because it consists of more histone, right? Basic dye. Then the word chromatin was first given by Fleming. So usually the shape of the nucleus will vary. They may be spherical, oval, and or discoidal. So usually because of the central vacuole, it is been pushed. The nucleus is actually pushed to the corner because you have the large central vacuole for the plant cell. So in animal cell it is located in the center. So let's see the parts actually. So with the parts we'll try to discuss still more. So let's see regarding the nucleus. So in interface you can see the nucleus which has nuclear membrane, and if you see it has nucleoplasm, all those stuff. So it has spherical bodies called nucleoli, and there is one more so one nucleus per cell, but we have number of nuclei also will increase in syncytium and in xenocytic condition. Some of the cells will lack the nucleus also. So if you see RBC, so no nucleus. So that's why we call it as enucleated, like that. And coming to the first thing is nuclear envelope. So this is double membrane bounded structure, and the space between them we call it as perinuclear space. We have the outer nuclear membrane. So outer nuclear membrane, and this is inner nuclear membrane. And here we have the perinuclear space. And we have the nuclear pore complex. So here we have it is being rounded by the structure called annulus. So why this nuclear pore is bigger is, so mRNA, proteins, rRNA, tRNA has nucleotides, enzymes that can pass through this pores, and you can see this fluid part which is nucleoplasm. Is clear fluid actually, in which only we have the chromatin material and nucleolus. So nucleoplasm is well distributed with cytoskeleton because to maintain the structure. And next here we have the nucleolus. Coming to the nucleolus, it is what discovered by Fontana, and it was appear spherical shaped body, and there is no membrane here. It disappears during cell division and reappears during telophase, mainly for production of rRNAs, means mainly for the production of ribosomes. Chromatin material, this will form a network. It consists of DNA as well as histone proteins. So these are the colored threads. So this will form nearly 2 meters long DNA distributed in 46 chromosomes. So 2 meters long. And coming to the function of nucleus, this mainly helps in the transmission of genetic material from one generation to another. Development, growth, behavior, reproduction. So it mainly regulates the metabolism, and it controls the cell differentiation also. Finally, let's see regarding chromosomes. So it is actually loose, indistinct network will be there. So we have different stages of cell division. So shows the number of structured chromosomes in place of nucleus. So coming to the morphology, we have different shapes and sizes of chromosomes. We can see very clearly during the metaphase. So coming to the haploid, the single set of chromosome we call it as haploid. N. 2N means by the fusion of two gametes, the diploid, the zygote will be formed. In that we have diploid 2N. So this we call it as genome. And this is zygote, formed by two gametes. So union of two gametes gives rise to zygote. 2N. So N plus N is 2N. So coming to the two very important words, that is karyotype and ideogram. So coming to the karyotype and ideogram, so revealing this number and shape of the nucleus by seeing under the microscope, we call it as karyotype. Number and shape. And here in this case, so the diagrammatic representation of the chromosomes in decreasing length. So in decreasing length, we will be calling it as ideogram. So it is diagram. So you'll see the diagrammatic representation by decreasing length. So coming to the ideal chromosomal structure is, chemically it is made up of DNA plus proteins. So here it is heredity material. Here it is histone proteins. So histone also contains again H1 linker histone. This is linker histone. Next we have H2A, H2B, H3 and H4. This will form the histone octamer. So in metaphase you can see clearly what is the structure. So let me show you the structure of chromatid. So this we call it as spindle fibers. And here we have the centromere. This is centromere. This is primary constriction. And this is secondary constriction. This is SAT chromosome. The relationship between these two are sister chromatids. And here we have a proteinaceous extension called kinetochore. And this part we call it as telomere. So let's discuss regarding this thing. So chromosomal structure, it is usually linear. It has two thick chromatids. We call that as two sister chromatids held together by a centromere. So chromatid is actually condensed to form a single chromatin thread. So because it consists of DNA. Apart from this primary constriction, it has secondary constriction also. The secondary constriction is the site where the nucleolar organization will be there. So it will formation of nucleolus at the end of cell division. A part of chromosome beyond the secondary constriction we call it as satellite. So the chromosome name we call it as bearing satellite, we call it as SAT chromosome. So because we call it as SAT means sine acido thymonuclein. So without thymonucleic acid, because of the relative deficiency of DNA in this part, you don't have the thymine in this part, and you have more guanine and cytosine. So that function is not yet understood, but the terminal regions we call it as terminal ends, we call it as telomeres. So telomeres maintain the integrity and individuality actually. So usually the ends of chromosomes are degraded by nucleases, which causes aging and senescence. So to prevent this only, eukaryotes contain repetitive non-coding sequence of DNA with proteins called telomeres. So it is continuously dividing cells on cancer cells. Telomerase replaces the telomerases. The telomerase replace telomers and prevent the aging. So let's see the various classification of this chromosomes. So this is metacentric. This is submetacentric. This is acrocentric. And this is telocentric. So coming to the telocentric part first, this is placed at the end of the chromosome. The centromere is actually placed at the end of the chromosome. This is called I-shaped. And this is the acrocentric chromosome. The centromere is slight away from the end, just slight away from the end, and this we call it as J-shaped. So J-shaped chromosome. And in submetacentric we have one long arm and one short arm. Long arm we call it as P and short arm we call it as Q. This is L-shaped chromosome. And metacentric we have a V-shaped chromosome, which is center, exactly at the center the centromere will be there. And you can see this shape during anaphase actually. In higher animals, the chromosomes can be differentiated into two types, like autosomes, allosomes. Like 22 pairs of autosomes we have. And remaining X and X is allosome, or X and Y. So totally 23 pairs. So 22 pairs of autosomes and 1 pair of allosome. So in the chromatin also we have two categories, euchromatin, heterochromatin. So euchromatin is actually transcriptionally active. This is transcriptionally inactive. And this is lightly stained. And darkly stained this one is. It is located more in the center. It is located in the periphery. So replication takes place early. So replication is late.